Hey guys, Dr. Brown here with another fascinating video about the human body and histology, and in this case we're going to be talking about the protein collagen. Now collagen, in addition to being the most abundant fiber protein in the extracellular matrix of connective tissues, is also the most abundant protein in the human body. You are in fact about 30% dry weight collagen alone. So if we were to take all the water out of you, 30% of what's left would be collagen. Collagen is actually not a single individual protein, it is a family of proteins, and we're going to be talking about several of the members of this family of proteins. And collagens come in three varieties: fiber-forming collagens, fiber-associated collagens, and network-forming collagens. And these fiber-forming collagens are the ones we're going to spend the most time on because these are some of the strongest biological structures in nature. In fact, the collagen from animal tendons, what we call sinew, is so strong that it was used to form the torsion springs in ancient siege weapons like catapults, which, we can admit are the inferior siege weapon. The most important fibril forming collagen, and indeed the most abundant of all the collagens, is what's known as type 1 collagen. Now, type 1 collagen actually starts out as three individual monomers, which then form a heterotrimer known as tropocollagen. This is about a 90 nanometer long protein which will spontaneously assemble, integrating in a 6-wide fibril called the collagen fibril. So as a note, just when you see fibril, note that that means it's something small that's then going to be assembled or bundled together to make a larger fibrous structure known as a fiber. You can notice some things about these fibers. There's a gap in between individual tropocollagen trimers. My thing is acting up really bad today. Okay, here we go. So here you can see there's a little gap right there. Now, because these gaps will occasionally occur on both edges of the collagen fibril, you'll notice that the fibril kind of dips in, right? The fibril's coming along like this, and it dips in, and then comes back up, and the same thing happens over here. And when viewed in the electron microscope, you can see these gaps in the collagen fibrils here and here, etc. So you get this very distinct striated appearance in the electron microscope. Now, collagen fibers can contain varying amounts of collagen fibrils depending on the thickness of fiber that's needed in any given tissue. Let's look at a few other collagens of note before we wrap up this video. Type 2 collagen is found primarily in cartilage, and type 2 collagen is going to form a much less dense network. It's going to form these looser fibers, which are going to then have space filled in with substances that we normally associate with the ground substance, like these proteoglycan aggregates and fibronectins. And this space is all going to be filled in here by these highly glycosylated compounds, which are then going to attract water, and the combination of the loose type 2 collagen fibers and the extra water in the space between those fibers is what gives cartilage its kind of special springiness. Now, there are some other fibril forming collagens that aren't the bulk of the fiber, but instead are going to help integrate with the type 1 and type 2 collagens to stabilize the network. And those are types 5 and 6. So type 5 collagen integrates with type 1, and sorry, that's not a 6, it's an 11. Type 11 collagen is going to integrate with collagen type 2. A couple other collagens of note that are really interesting and important for us as we continue to study connective tissues are the type 3 collagens, which form reticular fibers. And those are these dark fibers that we can see in here. These fibers appear dark in this light micrograph because it's been stained with a silver containing dye. So type 3 reticular fibers are what we call argyrophilic, meaning they have an affinity for silver. These reticular fibers are going to be found primarily in expansible tissues, like, say, the walls of an artery. And these, along with elastic fibers, which we're going to learn about in a little bit, and type 1 collagen, are going to give the artery wall a mixture of strength and elasticity, so that when that artery is pressurized by the contraction of the ventricles of the heart, it can expand, but then once that pressure goes away, when the ventricles relax, the elasticity imparted by these three different components of the extracellular matrix will allow the artery wall to return to its original size. And then finally we have type 4 or network forming collagen, and I don't have a good picture of that. It's not visible in the light microscope, and even in the EM it's not very impressive. But what's interesting about type 4 or network forming collagen is that this is the collagen that makes up the primary components of the basal lamina. And so as you might recall, the basal lamina is secreted not by fibroblasts down in the reticular layer, but the basal lamina is secreted by the epithelial cells above. So this is the only collagen that is abundantly secreted by epithelial tissue instead of connective tissue like all the rest. So that's our little overview of collagen. I hope you had a good time. Study hard, have fun. See you later.